Hey everybody. I'm still here in West Virginia. It's still cold. Went to the football game. UCF lost. This is terrible. So I figured, what do you do when you're depressed? You come and you make videos for your students talking about active and passive transport. So this is from last chapter, but students seems like I had some questions about this. So I just wanted to make a quick little video explaining how we do membrane transport. So what are we talking about? Remember, we have a membrane that is around the cell, and that membrane is made up of phospholipids, and phospholipids have a hydrophilic head and a hydrophobic tail. And what happens is they kind of align like this into the phosphobilipid layer. And hopefully at this point, you have studied um, the functions of the phosphobilipid layer and what it does. There are many different functions of it. So make sure that you understand all of those functions and you can name those functions. So now, since it is a phospholipid, it's still a lipid. So there are some things that can directly diffuse through the membrane, and that is going to be small fatty substances. They're going to be able to go through, like things like your thyroid hormone are going to be able to pass directly into that membrane. Other things are small molecules like CO2 can come out of it and O2 can go in it. Now, if you have something that's charged, that is going to be something that is polar. It's not going to want to go through a non-polar membrane. So stuff like potassium, or potassium is going to be on the inside. Stuff like sodium is going to be on the outside. It wants to get in, but we're going to have to use channels to be able to get that through. Um, so that's essentially what we're talking about is membrane transport. Like for example, when we eat food, we have our small intestine. And what lines our small intestine? Well, remember that is the simple squam, or not simple squamous, simple columnar epithelium with all those microvilli, right? And what do we have underneath that? So imagine that this is making your lumen. So this is like your small intestine, and it's come from the stomach. And what we need to do is we need to break all that stuff down into its constituent parts. You have your starches that have been broken down into glucose. You have your big proteins that have been broken down into their amino acids. And where do those need to go? Those need to go into us. They need to pass into us. Well, how do we get it into our bloodstream? Well, remember, these epithelial cells have all these tight junctions, and they got those desmosomes, and they have the anchoring proteins, the hemidesmosomes. So stuff can't pass through in between the cells. It's got to go in through the cells, and then the cells push it out the other side. So this is what we're talking about when we're talking about membrane transport. Okay, so there are several types of membrane transport, but we can put them into two relatively just simple categories. We have what's called passive transport and active transport. So you should be able to tell me the three different types of passive transport, and you should tell me the two types of active transport. And then you should be able to define passive and active transport. And all that means, passive and active, is one uses ATP, one doesn't. Passive does not, no ATP. Active transport needs it. So why would you need ATP? Well, this comes back to understanding gradients. So remember what a gradient was. Either you can have a concentration gradient, you can have a temperature gradient, you can have a pressure gradient, you can have a chemical gradient, you can have an electrical gradient. All that means is that there's a difference from one side to another. So you can think of it a bit like this hill right here. So you have a hill like this. And there is a difference between this height and this height. So what things like to do is they like to go down their concentration gradient. That's how stuff wants to move. Remember, the universe is in motion. Everything is in motion in the universe. Even this table that I have underneath me, these particles are moving, but very slowly. Eventually, they'll all fall apart at some point. So, how do they know how to move? Well, the rules of the road of the universe are things move from areas of high concentration to low concentration. So, in this case, with passive transport, we're going to have things that are going to go down their concentration gradient. So, and it doesn't require any energy to go down the hill. So, it's going to be diffusion, facilitated diffusion, and then osmosis. And I'll explain each of these individual ones. There's also filtration. There's also filtration, and that's how we get stuff in our blood into our, or stuff out in our blood, like glucose and potassium and sodium and uh, different things. That's how we get it out of our blood and or into our tissues, because our tissues and our cells need that stuff. So the main function, or the main thing that you need to take away from this, is that things in the universe move down their concentration gradient, and they don't require any energy to do that. If it doesn't require energy, that makes it a passive transport. Active transport, on the other hand, at some points, we're going to need energy to organize things. So we're going to have to, like, everything in your life tends to move towards being disorganized. Like if you come to my office, my desk, there's papers everywhere. When I'm in a hotel room right now, and half of my stuff is like all over the room. It's because things were packed tightly in a high concentration in my suitcase, and now they're spread out all over the place where they're not concentrated. Well, sometimes we need to organize things in the body. So when I check out here, I'm going to have to take all my stuff in my suitcase and put it back in. That's going to take it from an area of lower concentration on the floor back to an area of higher concentration into my bag. And that's going to require energy from me. So sometimes in the body, we're going to have to do that stuff to set some things up. And we're going to go over tons of examples through anatomy and physiology. So that is active transport, is when it's going up the hill, and it requires energy to walk up the gradient. Okay, so that's the two types of membrane transport. And then there's going to be, it's called active transport, like your sodium potassium pump is an example. But then there's also other active transports, like vesicular transport, like when we do phagocytosis and pinocytosis, which are endocytosis, and then we get rid of things out of vesicles, and that's called exocytosis. That's usually going to require energy to do that. All right. So we looked at these: passive, no energy required. Diffusion is the first one, simple diffusion. So for example, if you have oxygen and it is in a higher concentration on the outside here, and it's at a lower concentration on the inside, which way does oxygen want to move? It's high up here and it's low in here, so it wants to run down its concentration gradient. It's going down its concentration gradient, so no energy is required. That is simple diffusion. In facilitated diffusion, now some students will get messed up with this word and they're like, facilitated, that sounds like energy. It's not energy. All it is that you're using a channel to facilitate the movement of particles across the membrane. Because say this was sodium here, sodium is charged. That makes it polar. This inside here is fat. It can't pass through this. It gets deflected off. So it's got to go down. And it's still high on the outside here, this sodium, right? And it's low on the inside. This is probably glucose here, but it's low on the inside. And so it wants to go down its concentration gradient, but it can't get in because this membrane is stopping it. So it can pass down through a membrane or through a protein or a channel, and that is going to be facilitated diffusion. The last one is water. Water is one of the most important things that we have in the body. There's a lot of us is made up of water, like 50-some percent, 60% of us is water. As a nurse, one of the most important things you can do as a nurse is to make sure that the ins and outs are correct. Water flow should be in only the right spots. It shouldn't collect in areas. When it's collecting in areas, that's bad. When people are dehydrated, that's bad. So it's like my aunt always says, she's been a nurse for about 50 years. She would say, when some people would come into her ER, she's been working in Mount Sinai ER in Miami for the last 50 years. She said, Chris, you know what? She's from West Virginia. She said, Chris, I'm not making fun of anybody when I do this accent. My family they are all country. She would be like, Chris, some people come into the ER, they just thirsty. And what she means is you give them an IV and most people get better. A lot of people. And so being a nurse, understanding the way water flows is really important. So this is not just for this chapter. This is for all your other chapters. This is when we get to our kidney chapter. This is when we get to there's a whole chapter on water and pH. And and electrolytes. So understanding osmosis, I spend a little time and make sure that you get this. And we'll talk about that. It's just movement of water down its concentration gradient, which is a little weird. Active transport, we have active transport, we have exocytosis, and we have endocytosis. This is vesicular transport. This is just moving things in little bubbles in and out of cells. That's all it is. Okay, simple diffusion. So let's do an example here. You drop some red dye into a beaker with water. At first, it starts off really concentrated in the drop. When you drop it in, it starts to spread out. Now, over time, what's going to happen? All of these molecules are going to go from this area of high concentration into an area of lower concentration, and they'll spread out, and eventually the dye will look all red. We can do this experiment at home. You can take some food coloring, put it in a glass, come back 10 minutes later, all the molecules are spread out. So let's do another example of this. Let's just say, what room are you in? They're all at almost a 6-minute walk. Hold on one sec. So if you look, we have a beaker here. And imagine this beaker is split by a membrane. And we dump, imagine this is tea, right? And in this, we're going to make sweet tea on this side. And we're going to put a lot of sugar in here. So we have all these sugar molecules in here. And all these sugar molecules, what's going to happen is if we left this and we have side A and side B, what is going to happen to this if we leave this? Which way are the particles? Do they want to move? They're going to want to move to the area where they're less concentrated. So you come back, and if you originally tasted this sweet tea, it's too sweet. This tea is unsweetened tea. All of a sudden, now you come back, and they will become of equal sweetness because all of these particles are going to move over to this side until you get equal amounts, and they get to equilibrium. That's simple diffusion. There are some factors that are affecting simple diffusion, and this makes sense if you know that diffusion is just movement of particles. So what is temperature? Temperature is just it's the speed limit of stuff. So if something's cold, it's moving slowly. So the particles move very slowly. If it's warm, things are going to move faster. So temperature goes up, motion of particles goes up. Temperature goes down, motion of particles to go down. The larger the molecular weight of a molecule, the longer it's going to take. And that makes sense. Something bigger is going to move a lot slower. So you have imagine we're going to do a race here between this particle and this molecule. So we set a timer for 5 minutes on this race. And we say, all right, guys, see how far you can run. And we come back 5 minutes later, and A is here, and B is all the way over here. Which one is the larger molecule? And we would say that A is the larger molecule. Why? Because look how slow it was. It took forever just to get right here in the same time that B got down here. You can think this is like an offensive lineman running here, and this is like a speedy running back. Who's going to run faster? The smaller guy. The steepness of the concentration gradient is also going to make a big difference. So if you think of, okay, so you can see the other thing is the steepness of the concentration gradient. So here you have, this would be a very shallow gradient. That's not very steep. So how fast are things going to move on this? They're not going to move. But if you have a concentration gradient where it's super high on one side and super low on the other, it's going to be very fast how it's going to go down. And then obviously the more surface area you have, the more places, more doors, like the bigger surface area, the more doors the particles can go through, so the faster it's going to do that. Okay, facilitated diffusion, same thing as diffusion. The only thing is they can't pass through the membrane. So they've got to use a molecule, or they've got to use a protein or a channel to be able to pass through. That's all it is. Same thing, same rules of diffusion. Okay, now quickly with osmosis. Now, the definition of osmosis that the book and stuff uses is that water moves from regions of higher concentrations to lower concentrations. That definition sucks. I don't like that definition at all because when you think of concentration, water is usually considered the solvent, and things are dissolving in the solvents, the solutes. So you're usually thinking about the concentration of a solute. not the concentration of a solvent, which is so it's like counterintuitive in our brains to think that way. So I don't like thinking that. The way I like thinking about it is with parties. So, how do you know where a good party is? Well, you know where a good party is where all the people are. And water is a party animal. Water loves to go to parties. And so where does water want to go? It wants to go where the party is. How do you know where the party is? That's where the people are. Who are the people in this? The particles. So for example, and the students can get confused on this because you can get confused on diffusion here. So don't confuse this with diffusion. What's the difference between diffusion and osmosis? Diffusion is the movement of particles. Osmosis is the movement of water. In this case, let's just say that this membrane was permeable to these solutes in here. What would happen? We would have side A and we would have side B. If this was permeable, what would happen? The solutes would go over. But in this case, this membrane is too small. It's too tight. The only thing that can fit through it is water. So what happens is water, where does it want to go? It wants to go to the party. Which side has the party? Where the particles are, where the people are. So water goes, well, I want to go over here. And it's going to pull water. So you can think of it, water's like a little lost puppy and it's following everybody around. It generally wants to follow sodium around. So wherever sodium goes, that's where water goes. Think about it. You ever eaten something really salty? You gone to somebody's house and they like drop the meat in like a salt pile? I mean, not really, but you feel like they did, and you eat it and everything gets all swollen. Why? Well, because you have all that salt in you. It's absorbing, it's reabsorbing the water into your body. So in this case, we can see that water wants to go where the party is at. And another way of stating that in just a kind of a less fun way is that water moves towards a higher concentration of solutes. I like going to the party better. Just me. Okay, so hopefully you understand that. So this is requires no energy. It just moves um, down its concentration gradient. So in this case, water is going to move where? It's going to move to this side because that's where the party's at. The reason the solutes don't move over is because this membrane is impenetrable to them. The holes are too small. Okay, so now the last thing that I want to do before I have to go is the last passive diffusion part, or sorry, not passive diffusion, the last kind of part of active it's not active. I'm sorry. I'm like thinking about having to go. The last part of passive transport is this tonicity stuff. Now, what does tonicity mean? Tonicity is talking about the solution. So when we do this, I don't want you to tell me that the inside of a cell it has a tonicity. It's the solution is always the tonicity. And what is the tonicity? Tonicity is the ability of a solution to affect the size of a cell. And how does it do that? Through fluid movement. There are three types of solutions. There's a hypotonic, there's an isotonic, and there's a hypertonic. Well, why is this important? Well, when you go to the hospital, God forbid, and you're one of those patients that's just thirsty, right? You got something wrong with you and you're dehydrated. What are they going to give you when you go to the hospital? They're going to give you an IV, right? But do they hook you up to like a faucet and then just start dripping in water in there? No, they don't. What do they give you? They give you saline. Now, what is saline? Saline is salt water. It's a 0.9% solution of salt water. Why do they give you salt water? Are you a animal out in the ocean? No. It's because there are dissolved substances inside of our cell, and this is going to have to do with osmosis. So there are three types of solutions that we have. Hypotonic, what does hypo mean? Hypo means low. So let's do this example real quick. And this is not the way it works, but we're just doing an example real fast. Imagine that there are 4 particles inside of our cell. Now, you can memorize this if you want, but I think this is a better way to do it. If you have a hypotonic solution, which means what? The solution has less dissolved particles in it. So how many dissolved particles? Let's just say 2. And we're just picking that number because they're less. Okay, an isotonic solution. What does iso mean? It means same. So if we have 4 dissolved particles inside of the cell, how many dissolved particles are you going to have on the outside? 4. Okay. Now, in a hypertonic solution, what does that mean? That means elevated of the solution. Remember, you're never calling the inside of the cell tonic anything. It's just the solution. So there's four inside. And again, I'm just picking four because it's easy. How many is going to be on the outside? More. So we need five or more. So let's just draw a lot. So this one would be like fresh water. This would be like salty, salty water, like the ocean. And then this would be like a bag of saline, 0.9%. Okay, so now knowing what we know about osmosis, what do we know about osmosis? We know that it likes to go to the party. Where is the party? Where there are more people. It always wants to go. So in this case right here in our hypertonic solution, where is there a higher concentration of things? Where are there more people? In the inside. So which way does water want to go? Inside. Okay, now it's on the inside. If you start filling that cell up with stuff, what's going to happen to it? It could burst. That is called to lyse. Lyse. Cell lysis, hemolysis. If this is a red blood cell, hemolysis. Now, in the case of our isotonic solution, what is going to happen? They're the same. So water goes in, water goes out. Easy come, easy go. No water change, no balance. That's why you get normal saline when you go to the hospital and you need fluid, is because you don't want to blow up your cells. Then what happens? And you get a hypertonic solution. So what happens in a hypertonic solution is what? Where's the party? It's on the outside. So water goes to the outside. And it shrinks the cell. This is called crenation. Crenates. This is neither one is good. That's bad. Imagine this happens to a brain cell. That's bad news. Imagine that happens to a brain cell. That's bad news too. You can see this in action if you've ever been cooking. Say you like to cook steak. Sorry, my vegans. This is just an example. But say you're cooking a steak and you have the steak here and you're supposed to salt a steak prior to putting it in the pan. You put salt on it. What's going to happen to it when you put salt on the outside of a steak? If you left it there, you come back, what would you see? You'd see water coming out. It's going to draw the water out. And then what's happening? It'll dissolve the salt and it'll go in and it'll get in between the proteins and the myoglobin and all that stuff, and it'll tenderize the meat and it'll get deeper. It'll season it deeper into the meat, which is what you want. So now what happens here is that if you put so much salt on this, eventually what it would do is it would just dry the whole thing out, and then you would have dried meat. And the reason that they used to do this way back in the day is that's how you preserved meat. They didn't have refrigerators. So they'd throw a ton of salt on stuff. It would desiccate it. Bacteria don't like dry stuff. They don't like salty stuff, obvious reasons. What's going to happen? It's going to pull all the liquid out of their cells, and it's going to kill them. Unless they're a halophilic bacteria. So then what happens is this meat is all dried out. It's like beef jerky. And then you can keep it for forever. In fact, salt was so important back in the day, Roman soldiers got paid with it. And if you know what the word for salt is in Latin or Spanish, the word is sal for salt. And what do you get paid every day, every 2 weeks? You get your salary. That's where that word comes from. It comes from the salt that they used to use to dehydrate their meat and season it and all that other stuff. Okay, so just really briefly, make sure that you can do this with your red blood cell. Put a red blood cell in a hypotonic solution, isotonic solution, and tell me what happens and why. So remember, you're talking about the solution. You're not talking about what's inside the cell. And it's and then remember, water goes to where there's more particles. So in a hypotonic solution, there's less on the outside, there's more on the inside. Water wants to go where there's more. Same on the outside, same on the inside. Water goes in and out the same. It's an equilibrium. In a hypertonic solution, you put a cell in there, there's more dissolved particles on the outside. Water goes out. It shrinks. Okay.